Today we will be discussing replication in prokaryotes using the prokaryotic replication fork. Prokaryotes such as E. coli have a single circular DNA molecule which represents its genome. The cells undergo a process known as theta replication, in which the double-stranded DNA is unwinded bilaterally at the origin of replication. DNA is synthesized, in which there is the parent DNA strand along with the new DNA copy, which are eventually passed on to the daughter cells. Let's take a more in-depth look at replication at one of the two forks present. The model shown is that of a replication fork, where I have used Twizzlers to represent DNA strands and various colors of Play-Doh to represent the different structures that allow replication to occur. Replication occurs in a 5' to 3' direction, which causes the top strand to be the leading strand and the bottom strand to be the lagging strand. The green structure at the mouth of the replication fork is known as helicase, which breaks apart hydrogen bonds and therefore unwinds the DNA strand. The yellow structure to the far left is known as gyrase, which relieves built-up pressure from supercoiling. Single-stranded binding proteins are responsible for keeping the DNA single-stranded, in that they do not coil or return to the original state. This is represented by the orangey-yellow molecule on the leading strand. RNA primase is represented by the blue molecules and holds a primer that binds to DNA polymerase and allows it to function. DNA polymerase III binds to the primer at the RNA primase and adds nucleotides in order to replicate the strand. The leading strand is replicated continuously, whereas the lagging strand is replicated discontinuously. This discontinuous replication is responsible in creating small segments known as Okazaki fragments. Once this occurs, DNA polymerase I is then used to get rid of the RNA primers and replace that with DNA, as shown in this demonstration. The last structure shown in this greenish-red molecule is ligase, which attaches the DNA to phosphodiester bonds in order to completely seal the strand. The major purpose of the 5' to 3' exonuclease function of DNA polymerase I is to remove primers used during replication and replace it with DNA. If this function were unable to perform, I would hypothesize that Okazaki fragments would be unable to connect, along with the DNA not connecting to phosphodiester bonds. These are the references that I have used. Thank you very much.